Hello, everyone, and welcome to No Waffle GCSE. Today, we're going to do some of the hardest science topics revision. So, we're going to start off with homeostasis and response. What I want you to do is you have 10 minutes to blurt as much as you can on the screen to help, and then we'll move on to the next topic, and that's how it should work. I'll put the instructions up, and let's get going. If you have any questions, do ask. Just to say, I think that my mic wasn't on earlier, so I'm sorry about that. But we have now moved on to ecology. So blurt as much as you can using the prompts on the page. So hopefully you have had time now to go through the entire of this topic. We're now going to go through and see if there's anything we missed in our blurting. So let's begin with communities. This is definitely not enough information, I think. So more information could be added to the communities. So what you want to say is, as an individual is part of a species, it then lives in a habitat within a population. So what we're saying here is that there's a population, and within this population, we then have different types of habitats. And within these habitats, we then have different species. And when there are many different populations in the same habitat, then this creates a community. So let me just rub that out. So a community are loads of different populations living together. Community with the non-living parts of it. So we're talking about an ecosystem. It's the interaction of a community with the abiotic parts of its environment. So let's look at some of the abiotic factors and the biotic factors. So abiotic include light intensity, temperature, moisture levels, soil intensity, we forgot, direction, carbon dioxide levels, and finally oxygen levels, especially for aquatic organisms. Biotic would be new predator, new prey, new pathogens, competition, as well as food availability. Now, in terms of the adaptations, because that was the next one. So we have structural, not aesthetic. So we have structural would be the shape or the color of something. For example, the ability of it to camouflage. Behavioral is the way an organism behaves. For example, pretending to be dead to avoid predators. And functional will be involved in processes such as reproduction and metabolism. So, for example, late implantation of embryos or conservation of water through the produce of sweat. Now, here is where we should have discussed extremophiles. Because extremophiles are organisms that live in extreme conditions. For example, high temperature, high pressure, or high salt concentrations. Now, an example is bacteria, which live in deep sea vents when the pressure is variation for different scenarios. So, which we're looking at some examples there. Now, the next one was levels of an organism. So this has required you to say stuff like, it all begins with the producer, and then you have the primary consumer, the secondary consumer, be at the top. So Now, we looked at the carbon cycle and the water cycle here. And then decomposition. So things that affect the rate of decomposition are the moisture levels, because microorganisms grow faster with water as it's needed for respiration. Temperature, chemical reactions tend to work faster under temperature, and it's enzyme controlled. And also availability of oxygen. Now, compost is where biological matter has decayed, and it proves this. So, decayed, broken down state of living organisms. It's used by gardeners and farmers as natural fertilizer to provide optimum conditions for decay, so oxygen and increased temperature. Methane gas. So microorganisms decompose anaerobically, as we saw here, to produce methane. Now this can actually be burnt as a fuel. So this is why we didn't take this further enough. So we said methane can then be burnt as a fuel. And as it's burnt as a fuel, you then have your biogas generators. And these are used to produce methane. So biogas generators produce methane with all the optimum conditions so that they can be burnt as a fuel. And then we have the practical for the decay of milk. So we can go through that now. So we start off with you add your volume of milk. You then add the indicator phenolphthalein. And in order to make this colour change to pink, you need to add an alkali, which is sodium carbonate, for example. So now you know that once it starts producing the fatty acids as the lipid breaks down, then you will see the change in the phenolphthalein. So we have milk that's initially quite white, and then it turns into pink as you add the alkali and then the indicator. So we now have pink. Now what you would do is then you would add the lipase enzyme, I believe. Is it lipase enzyme? Yeah. You then add the lipase enzyme and you start the stopwatch. And you see how long it takes for the solution to turn colourless. Because this tells you how long it takes for it to produce fatty acids and therefore how long it takes for it to break down, because we know that lipids break down into glycerol okay. Now, environmental changes can actually affect the distribution of species. For example, temperature. Climate change may lead to insects migrating to places in the world where they are becoming hotter. Water availability. Populations migrate to find water. Atmospheric gas composition. Certain pollutants can affect the distribution of organisms where sulfur dioxide is created. Now, these changes may be seasonal, geographic, or caused by human interaction. Biodiversity is the fact that species are less dependent on each other for things. Now, most human activities are having a negative effect on biodiversity. The future of humans on Earth depends on maintaining biodiversity, for example, food and new medicines. Now, more land is being used for housing, farming, shops, roads, and flats. Pollution kills animals. The sewage, fertilizer, and toxic chemicals can pollute the water. Smoke and acidic gases can pollute the air, and landfill is quite toxic and can result in the pollution of land. Now, if we can also talk about the eutrophication, which is a process by which the water becomes really bad. So, eutrophication. So it starts like this for eutrophication. There's a nutrient leakage in a water body. Then what happens is because of algae grow. Now eventually after the algae grow, there will then one day not be enough. There will not be enough nutrients to sustain the algae because they would have used it all. So then there's no more nutrients left. And then the algae starts to decay. And as it decays, it then means that are respiring. And as microorganisms respire, they release carbon dioxide and use energy, use oxygen. This then starves the other sea life from oxygen and can actually cause deaths in populations. So that's the process of eutrophication. Now let's look again at peat bogs, because that's where we got up to. So peat bogs are a type of habitat, and peat is a material that forms when a plant hasn't fully decayed because there's a lack of oxygen. So that's the point which we missed. So peat is formed when it's not fully decayed due to the lack. It then accumulates within the bogs. It's quite acidic and waterlogged, and these bogs are a habitat for many different species, for example, migrating birds, and as they are destroyed, they're being drained in order to create space for farming, and peat is often used as compost. Sometimes even the dry versions are used as a fuel. Now, deforestation is the cutting down of a large number of trees to have land for cattle and rice fields and grow crops. Okay. Now, another thing is that we need to talk about is biomass. How is only 10% of biomass passed on to the next trophic level? Why is that? Well, firstly, not all biomass can be eaten. Carnivores can't generally eat bones, hooves, claws, and teeth. Not all biomass eaten is also converted into biomass for the animal eating it, because glucose is used in respiration, excretion, urea is waste substance, biomass consumed can be lost as feces, things like that. Food security and factors affecting food security here. Increase in population and increase in quality life, because of the changing diets. As more countries are becoming developed, more food resources are therefore being used. New pests and pathogens can also destroy crops, especially ones with little variation. And then conflicts in some countries that can affect the availability of water. So the farming techniques of raising them in small cages, where there is food, so it's a very manufactured lifestyle. Now, this type of farming has many ethical objections, and this actually causes stress. It also increases the increased risk of spread of infection, and it's carried out to increase profit and increase efficiency, but it actually decreases their quality of life. And is it fair to do this? Now, we also have sustainable fisheries. So, because of the fact that the number of fish in the oceans are decreasing, humans have to fish at a humans need to stop fishing at a faster rate than the population can regurgitate and breed again. So, limiting net size, have fish quotas, things like that. Biotechnology, for example, the fungus fusarium, which can be used to produce mycoprotein. This is a protein-rich source and is suitable for vegetarians, and therefore it doesn't require, which reduces land as well as main contribution. Genetically modified bacteria, such as how insulin can be taken and purified to treat people with diabetes, and genetically modifying crops to have certain properties can have many advantages. For example, modifying them to be resistant to pests or extreme weather conditions can increase yields. Modifying them to increase their nutritional value in beneficial places where they lack access to certain vitamins, such as golden rice. So that is it for ecology. Not too much information. So we have now completed two well done, and give yourself a pat on the back. We are now going to move on to organic chemistry. So let me just show you the slide for that. So here you can see that we have the blurting page with a couple of different prompts for you to blurt out as much as you can remember. Now, this is a much quicker topic, so hopefully we will be done with this topic by 6 o'clock, and then we will then be on track to where we should be next topic. You now have 10 minutes to write as much as you can and blurt everything you know about this topic. Hello everyone, sorry to interrupt you. I'm now going to go over organic chemistry. So on the whiteboard, if you've missed any piece of information, do write it down in another colour so that you know what you've done wrong, and also well done for getting this far. So let's look at organic chemistry. So the first thing is hydrocarbons. So hydrocarbons are purely just hydrogen and carbon, nothing else. That's a key part of it. Now there's different types of hydrocarbons. Now the most simplest form is called an alkane, and the alkane is an example of a homologous series. Now what we mean by a homologous series is a group of compounds that react a similar way. Alkanes have the chemical formula CnH2n plus 2. It's relatively simple. We have monkeys eat peanut butter. So we have methane, ethane, propane, and butane. Methane will look like this, CH4. Propane will have 2, so CC. Now, these alkanes are what we call saturated molecules. The reason why we call it saturated molecules is because we know from our previous studies in giant covalent bonds that carbon forms 4 bonds to fully react. Now here, in each of the alkanes we can see 1, 2, 3, 4, 3, 4, and then vice versa, 1, 2, 3, 4. So we can see here that 4 bonds are being created for every single carbon, no matter what. Therefore, we know that they're saturated, meaning they're fully reacted. Now, hydrocarbons can have certain chemical properties. Now we can look at them now. So, the chemical properties of hydrocarbons. First of all, flammability. Hydrocarbons can be used as fuels, so the flammability is important. And I'm going to do a column for the short chain hydrocarbons and the long chain hydrocarbons. So when there's a tick in the short chain, that means that it's better for short chain, and when there's a tick in the long chain, it's better for long chain. So flammability is much better for short chain hydrocarbons, which means smaller alkanes. For example, here you can see that methane is a shorter chain than propane, and therefore methane will be more flammable. So, flammability. The next property is volatility. So volatility is how low the melting point is, and short chain hydrocarbons have the lowest melting point. Now volatility is a good product because as a fuel, you don't want to have to supply so much energy just to get the energy back out. That's counterintuitive. And then we have the thickness and how gloopy it is. So is it runny or is it viscous? Now viscous means quite thick and gloopy. Now the shorter the chain of the hydrocarbons, the more runny it is. And then the longer chain hydrocarbons are more viscous and sticky. So, clearly we can see that the more preferred route for fuels is the short chain hydrocarbons because we can see that the properties are much more favourable in terms of if you wanted a fuel. So how do we solve this? How do we get rid of the long chain hydrocarbons in order to get the small ones? Well, that's a process called cracking. So cracking allows you to break down long chain hydrocarbons into short chain hydrocarbons. Now it allows you to do this, and there's 2 different methods. So the first one is catalytic cracking. This is when you have a catalyst and an oxide. And what you do with this catalyst is you then vaporize the vaporize the hydrocarbon, and then let it flow over the aluminium oxide, and then it would therefore separate on the solid into a shorter chain hydrocarbon. So alkanes can be broken down. This is a thermal decomposition reaction, by the way, so energy is added in. And alkanes can be broken down into shorter chain alkanes. But then something else is formed on the side, and we call those alkenes. Now let me return back to that in a second. The second type of cracking is called steam cracking, but you don't need to know too much about that. So let's return to this idea of the alkene. So the alkene is really interesting because this then creates a whole new molecular formula. Now, alkenes have the molecular formula CnH2n. And that's it. So it starts ethene, propene, butene, etc. And for ethene, you have 2 and then H4. And because the carbon needs to form 4 bonds each to be fully reacted, there's an extra bond here. So here we see a double covalent bond. Now, alkenes are not fully saturated. These are unsaturated because of the fact that it has to give an extra bond in order to be fully reacted. So they're unsaturated molecules. Now, what else do we need to know? Okay. Let's just do a zoom in to the alkene reactions before we return to fractional distillation of crude oil. So react with a number of things in reactions called addition reactions. Now, addition reactions can be really beneficial for alkenes. So if you react an alkene with hydrogen, this is called a hydrogenation reaction. And what would happen is the hydrogen would just add on to the carbon and hydrogen, to the hydrocarbon structure. Now we also have alkene water. Now alkene plus water can create alcohol, believe it or not. Alkene can also react with halides. Now, alkenes can react with halides, and a key reaction of alkene and halide is alkene and I think it's bromine, alkene and bromine, which reacts to produce the alkene plus the bromide at the end of it. Now, what this suggests is there's actually this reaction is specifically important because this chemical, I think it's iodine, actually, it might be iodine. It can be used to tell us whether something is an alkene or an alkane. Because what happens is it's a color change reaction when you react it with alkene. So when you react it with alkene, it goes from orange to colorless. But if you react it with an alkene, because of the fact that no reaction will take place, it should just stay orange. And that's how you can test whether something is an alkene or an alkane. Okay. Now, this reaction is probably the most important in this case because we're getting alcohol. Now, alcohol can be created also via a process called fermentation. And fermentation would just be reacting glucose so glucose plus oxygen creates carbon dioxide plus ethanol, and this ethanol is therefore an alcohol. Okay. Now, alcohols can have further reactions. So, alcohols can actually react with oxygen to produce a carboxylic acid. Now, just so you know as well, as we said, different things form different homologous series. Now, homologous series is very similar to functional group. Functional group is the structure of that group, and it's a structure of all the different homologous series. So, in terms of an alkene, alkenes one would just be this single bond. An alkane, sorry, that is an alkane. An alkene would be this double bond. Whole would be COO, and then I think it's alcohol with hydrogen. We're going to double check this, but then you get the carboxylic acid is like this. Let's double check that information for you. The alcohol. No, we were originally right actually. It was alcohol plus oxygen makes that functional group. So have we discussed the functional group of the alcohols? Yeah, okay, we have. That's fine. Now we have something called esters. Now esters can come to place when you react a carboxylic acid with an alcohol. And as you react the two, you're going to get something called an ester. And that's what we call polyesters. So, yeah. And they have the functional group COO. So I actually believe that alcohols has the functional group CO. Now we also have something called condensation polymers. So condensation polymerization involves monomers which contain different functional groups. And what happens is the monomers then react together and the bonds form between them. For each new bond that forms, a small molecule is created. Now, I'd definitely say for this topic, it's all about practice. So try to ensure that you get as much practice for condensation because that's definitely the tougher topic. Now, some naturally occurring polymers here. So firstly, we have amino acids. So amino acids have an amino group and a carboxyl group. So amino acids contain two different functional groups, a basic amino group and an acidic one. Now what then happens is that you have Okay, so an example of, so an amino acid is a combination of the amino group as well as the carboxyl group. So that's why it would look something like this. That's the amino group. And then the carboxyl group, we know as this, that. And then you can connect it, for example, like that. But we will be doing some chemistry, quantitative chemistry, as well as some practice like this furthermore. We also have DNA. Now, amino acids can form polymers known as polypeptides via condensation polymerization. Now, the amino group of an amino acid can react with the acid group of another and so on to form a polymer chain. So, for every new bond that is formed, a molecule of water is lost. Again, we'll continue to practice that. So that is organic chemistry done. Now let's move on to the next topic, which is chemical analysis. Again, this is a really small topic. We're just going to be focusing on the numbers 3 to 5, so the tests for gases, anions, cations, and identifying ions. So if you can just blurt out all of the tests that you can remember, and I will respond back to you within roughly 3 to 4 minutes. Okay, let's do some revision of chemical analysis. So first we're going to begin with tests for gases as suggested here. So tests for gases and anions. Let's start with tests that we normally need to test for are chlorine, hydrogen, carbon dioxide, and oxygen. So let's do the tests for each of them. So for chlorine, what you're going to do is you're going to do a damp litmus paper test. And if it does have chlorine in it, then it will be bleached white. Hydrogen. So for the hydrogen test, it's the splint test, sorry. And what you do is you have the splint, and it's a squeaky pop test. So you have the burning splint, and if you put it in the test tube which contains hydrogen, then a squeaky pop should be made. A squeaky pop noise should be made if hydrogen is there. So with carbon dioxide, you then have another test which you bubble it through lime water, because as you bubble it through lime water, it then creates a white residue, and that stays there. And then oxygen can be measured using, again, the splint, and you put it in a test tube, and you say that oxygen is required for the combustion reaction of fire. So the combustion reaction would have to stop if oxygen wasn't there. So if the fire continues, then you know that there's oxygen. Okay, that's for the test for gases. Now let's look at the tests for the anions. So, tests for anions. Firstly, we have the carbonates and the sulfates and the halides. So the things that we need to test: carbonates, sulfates, and halides. 3 things we need to test. So in order to test for the carbonates, what we're going to do is we're going to react it with dilute acid. And what should happen is the carbon dioxide should then be produced. And then if you add a couple of drops, if you then connect this test tube to a test tube of lime water, because we remember that the lime water tests for carbon dioxide, we then know that we can easily test whether the carbon dioxide is in there. Now with sulfates, we're actually going to be reacting it with, instead of a dilute acid, very strong acid, hydrochloric acid. And you also have something else called barium chloride. Chloride, that the purpose of the barium chloride is that barium sulfate will form. And barium chloride, the precipitate, which is the word I was trying to think of for that one. So the precipitate formed from barium chloride is actually white. So a white precipitate formed. And as a white precipitate is then formed, then this suggests that it has reacted. You can test with nitric acid as well as silver nitrate solution. And you should therefore then get silver nitrate, and depending on the halide, so you can get different responses. So you can get chlorine, or chloride, bromide, and iodide. So these get you different colors. You get white for this one. You get milk, so milk, and then cream for bromide, and then a more yellowy color for iodide. Milk, butter, milk, cream, and eggs. These are just the colors of the precipitate formed in each of these solutions. Now, in terms of tests for cations, so first of all, we have the flames test. And for the flames test, we have all of the different metals and all of the different flame colors. So let's first write the flame colors. So we know that there's going to be a crimson red. We know that there's going to be a green flame. And I think green is for iron. The crimson red is for lithium ions. Also, no, copper, not iron, sorry. I don't know why I put the iron symbol. So copper to be green. And then we know that sodium is yellow. Potassium is lilac. Which probably needs a bit more toning down, so just imagine that's lilac. And then calcium ions with an orange-red flame. So what you do is, in order to do the test, you have to have a clean platinum loop. You then place it through the flame, and then it should bring these flames. So let's just test ourselves on it again. So we've got sodium, potassium, calcium, copper, and lithium. So let's attach the colors to them now. So we've got crimson red for lithium. We've got green for copper. For calcium, we've got this orange red. So let's do this orange brown. For potassium, we've got lilac. And for sodium, we have got yellow. And these are the flame tests. But some can also be tested by reacting it to a hydroxide, and then it then forms a coloured precipitate. So some metals form a coloured precipitate when reacting to certain substances. Now, the coloured precipitates can be quite useful. So as long as you react it to as long as you react the metal to a specific hydroxide, which is represented by OH, then you can then get produced a coloured precipitate of that ion. So you get the metal ion, which can form coloured precipitates. So you do need to know the colours of them. So let's discuss them now. So you've got calcium, copper, iron 2 and iron 3, aluminium and then magnesium. I think calcium is white. Copper is blue. Iron 2 is green and iron 3 is brown. Aluminium is red, then becomes colourless, and finally magnesium is also white. So let's just revise that again. So we've got calcium, magnesium, iron 2, iron 3, copper, and aluminium. So iron 2 is green, iron 3 is brown, magnesium is white, calcium is white, aluminium is white and then turns colourless. Okay. Then just some revision on the evolution of the atmosphere. So the three different phases are what we can revise of the atmosphere. So phase 1 is the volcano eruptions, and the volcanoes then produced a lot of nitrate ions, so which caused most of the chemical composition to be nitrogen, not nitrate ions. So most of the composition was nitrogen. There was barely any oxygen, and there was carbon dioxide as well, and there were some other gases like methane. Phase 2 began to happen, that because of the carbon dioxide we then had plant growth, that absorbed in the carbon dioxide. Now the lots of carbon dioxide was removed from the early atmosphere as it dissolved in the oceans. This dissolved carbon dioxide then went through a series of reactions. So it was not only absorbed just by the plants, it was also captured in different rocks and then became fossils eventually. And green plants and algae evolved and absorbed some of the carbon dioxide so that they could carry out photosynthetic carbonates from the ocean. So there's a lot of carbonates from the ocean. Now, when plants, plankton, and marine animals died, they fell out of the seabed and got buried under layers of sediment. Things like coal, crude oil, and natural gas are then made by this process of fossil fuels. And then phase 3 was that these plants and algal systems all produced oxygen. Now, as they produced oxygen, this allowed organisms to respire. And the first plant to evolve was algae. Algae evolved first 2.7 billion years ago, approximately. And then over the next billion years, then at some point green plant also evolved. Now today, eventually about 200 million years ago, the atmosphere reached a composition similar to 1% nitrogen and 20% oxygen, and then other gases in the atmosphere too. Okay, now we're on to the final topic, which is all about using resources. So you can have a little break before we start, and then I want you to write as much as you can remember, blurting out this topic of using resources. Thank you for joining the live stream. I was just encountering some internet issues which is not letting me share the screen and all of the work that I've just been doing for the using resources. But I hope you managed to take something good out of this revision session of blurting, and hopefully I wish you all of the best of luck for this week's half-term revision, and try and make sure it's more productive than the previous week. If there's any questions, see you soon.